This video is on how to do the meiosis activity. We are completely taking it from this website at University of Pennsylvania, and these are the authors of it. And go to the website, read over the information. Most of this I talked about in lecture. So we're going to start with the procedure. Now, your cells have chromosomes. So here's some beautiful chromosomes I made with Play-Doh. This is chromosome number 1. This is the copy from mom. This is the copy from dad. So you get one chromosome from mom, one from dad. Here's chromosome number 2. You get one copy from mom, one copy from dad. So the chromosomes look like this right here. So again, this would be mom's in blue. This would be dad's. Now for mitosis, they replicate. They both are going to copy themselves, and they are going to divide. So you have two pairs, and then they separate, so each daughter cell gets one from mom, one from dad. So let's put our chromosomes into our cell. So here are our chromosomes, mom's, dad's, and this is chromosome number 1. Now, this is boring with just one chromosome, so let's put in a second chromosome. So we have our cell here. This is our cell. It has two chromosomes. It has one copy from mom of chromosome 1, one copy from dad from chromosome 1. One copy from mom, one copy from dad. So what you're supposed to do is you're supposed to put a large oval in the front of your table. Now I am using Photoshop. I'm actually using Paint Shop. And that is my cell membrane. So here's my large circle, and that's going to be my cell. Now this is our genome. We have 2 chromosomes. So what is our haploid number of chromosomes, and what's our diploid number? Well, haploid is half, so there's chromosome 1 and chromosome 2. So my haploid number would be 2. So I have 2 different chromosomes. My diploid number, the whole number of chromosomes, I have 2 pairs of, I have a pair of 1, I have a pair of 2, so the diploid number would be 4 chromosomes, or 2 pairs of chromosomes. Now, we are going to be adding genes to this. We're going to be playing with this thing. They used to call it a trifle on this website. They changed the name because it sounded too much like a tribble, which was a Star Trek character. So they're calling it a Sivinia. So we're going to put genes on the chromosomes. And we're going to put the gene for fur color, green and yellow. Here, we're going to put the gene for straight and curly here. We're going to put genes for eye color and eye length. We're not really going to do anything with the genetics, but I just want to put at least 2 genes on. So let's just put hair color and let's put the genes. So I put the genes on here. This chromosome codes for yellow, and that is curly. And then this is going to be green, and it is going to be straight fur. So that's that one. Then this one is going to be blue eyes and long eyelashes. This is going to be red eyes and short eyelashes. Now, since we're not going to do anything with the genetics part of this, it doesn't really matter what traits you pick or what letters, but just one of these needs to be a lowercase, one needs to be a capital. One of these needs to be a lowercase, one of these needs to be a capital. Again, here are my chromosomes from Play-Doh, and here are the genes. I taped these. It actually suggests writing on the Play-Doh with Sharpie. That never works. So the next step on the instructions is to replicate the DNA. That's what happens to start meiosis and mitosis. So we're going to replicate the DNA. Bam! I now have 2 copies. I have 2 pairs of chromosome 1. So here's one chromosome 1 from mom. Oh, here's the second chromosome 1 from mom. It replicated. Here's the second chromosome 1 from dad. Here's the second chromosome, or first chromosome 1 from dad. So the DNA has replicated. And that's happening during interphase. So here, this is what we started with. Yes, it's colorful. So here's 1. It replicated. So now we have 2 from mom, 2 from dad, 2 from mom, 2 from dad. So we're going to have something like this after replication. So each one of these replicated. So these are both chromosome 1. These are both chromosome 2. And yes, I'm about to cut and paste these. So 20 minutes later, I have my duplicated genes. And we have 2 copies of mom's chromosome 1, 2 copies of dad. So again, mom has a little g. That usually refers, that's a recessive gene. It doesn't always show itself. Dad has the big C. Mom, though, has the big, or G, has the big C, but dad has the little c. So, same here. Mom has all the big letters, dad has all the little letters. And again, we're not really worrying about recessive or dominant traits. We want to pay attention to what happens to these, because they're going to get reassembled in a second. So, question, how many chromatids are present? Well, count how many there are now. So, prophase 1. What's going to happen is they're going to get tangled up with each other. So in our Plato model, those guys are getting dangerously close to each other. Oh no, they crossed over. Oh no, they broke off and reformed. So that's crossing over. It happens in prophase 1, where little bits of DNA from one chromosome break off and reattach to the homologous pair. So this was chromosome 1, so I believe we're saying purple was mom, so part of mom's purple broke off, got onto dad's. Part of dad's broke off, got onto mom's. So we're going to do the crossing over for our 2 pairs. Now this is the centrosome, right there. It's not the body of a guy doing jumping jacks. And usually it happens far away from this point. So it's going to happen on the ends here, not right there. So I'm going to take a little bit of this one, break it off, a little bit of this one, and break it off. So now the parts are broken off, and they're going to reattach to the like ones. Ta-da! So now I've rearranged the G's. This mom or dad used to be big G and little c. Mom used to be little g and big C. Well, now I have 4 different ones. I have little g, big C, big G, big C, little g, little c, and big G, little c. So I have all 4 possible combinations. Now I'm going to repeat that on the next one. So the little bits of DNA broke off. No, I didn't do it very neatly. And they're going to reform. I just lost it on the homologous chromosome. There we go. So a little bit of dad's got on here, a little bit of mom's got on here. So again, mom was originally big B, big L. Now there's one big B, big L, little b, big L. Here we have big B, little l, little b, little l. So it's rearranging the chromosomes. So in metaphase 1, I should say, they are going to line up across the center, like so. And then in anaphase, they're going to move apart. So let us do anaphase. So they've moved apart to opposite poles. And then in anaphase, then we have telophase and cytokinesis, where it's going to divide in 2. So this is the end of telophase 1 and cytokinesis 1. So I have 2 cells. And these cells, they're not like the original cells already. We have mixtures. So this looks like it's 95% mom, but there's a little bit of dad right there. Same thing, 95% mom, but a little bit of dad. Same thing here, 95% mom and a little bit of dad. But I want to change this a little bit. Because I made mom go to mom, dad go to dad. We don't have to have that. So in reality, when it's split up, we could have had mom's chromosome number 1 go to this cell, but dad's chromosome number 2 go to this cell. Dad's chromosome number 1 go to this cell. Mom's chromosome number 2 go to this cell. So you're getting random distribution. Not only are the genes mixing up, but so are the chromosomes. So this is neither of the parental DNA now. It's now half mom, half dad, and vice versa. So we are now in prophase 2. Now in prophase 2, not much happens. What eventually is going to happen is the homologous chromosomes are going to all line up across the center, and you guessed it, metaphase 2. So in metaphase 2, they've lined up across the center, and the centromeres or the spindle fibers are going to go grab onto the centromere, and it is going to pull them apart. So for this one, I pulled it apart, and notice I put the ones that had crossed over on opposite sides. But on this one, I'm going to put the ones that crossed over on the same side. So Now they've completely separated, and I've mixed up chromosomes 100%. So these are not like any of the parental genes. So we've split up, and we're going to get, so we have all of mom there, we have some of mom with most of dad, all of dad here, most of mom with some of dad here, and vice versa over here. So what happens in meiosis is the chromosomes are getting all mixed up. The genes are getting mixed up, and that's called independent assortment, mixing the genes up. Now, lastly is telophase. And what happens in telophase and cytokinesis is these are going to divide, and we're going to end up with 4 haploids. So we end up with 4 gametes. None of the cells are identical. None of the cells have the exact same chromosome matches that the parental DNA. So this is mom, chromosome 1's intact, but we have bits of dad's chromosome 2 and a little bit of mom left over. Here we have chromosome 1 from dad with a little bit of mom, and then we have chromosome 2 from dad, or mom, and then we have a little bit of dad's there. So we have the chromosomes all mixing up. So go through the directions on here, and what I want you to do is take pictures along the way as you go. And again, this is completely stolen from these authors. This is not my activity. So where to get parts? You can use Play-Doh, as I did in my example. You could just go to the internet and print out chromosomes. That's actually what I was going to. So I literally went and searched chromosomes to print out. And I have my yellow-orange pair, my red-blue pair, my green-purple pair. Ooh, this one even has the genes on it for you, so you wouldn't even have to put the letters on. So you could just print these out. My printer was not speaking to me today. That's why I did it online. So that is what your project is.